The new formula that could save millions of lives is an experimental cancer drug called dostarlimab. This drug belongs to a class of medicines known as immune checkpoint inhibitors, which work by activating the body's own immune system to fight cancer cells. Unlike conventional chemotherapy, which directly attacks the cancer cells and often causes severe side effects, immunotherapy helps the body recognize and destroy the cancer cells without harming normal cells. Dostarlimab has shown remarkable results in a very small trial done by doctors at New York's Memorial Sloan Kettering Cancer Center. The trial involved 18 patients with rectal cancer, a type of cancer that affects the lower part of the large intestine and often requires surgery to remove the tumor and part of the bowel. The patients took dostarlimab for 6 months and then underwent a scan to check their response. To everyone's astonishment, every single patient had their tumor disappear completely. This means that they achieved what is called a pathological complete response, or PCR, which is very rare and usually associated with a better prognosis. The trial was published in June 2022 in the journal Lancet Oncology and has been hailed as a breakthrough in cancer research. Hannah Sanoff of the University of North Carolina's Lineberger Comprehensive Cancer Center, who was not involved in the study but wrote an editorial about it, said that this was the first time in history that anything worked in 100% of people in cancer medicine. She also said that this could be a paradigm shift for rectal cancer treatment, as it could spare patients from having surgery and its life-changing consequences, such as incontinence and shame. However, there are still many questions and challenges that need to be addressed before dostarlimab can be widely used for rectal cancer or other types of cancer. For one thing, the trial was very small and only included patients with a specific genetic mutation that makes their tumors more responsive to immunotherapy. This mutation is found in only about 5 to 10% of rectal cancer patients, so it is not clear if dostarlimab would work for the majority of patients who do not have this mutation. Moreover, the trial did not have a control group, which means that there was no comparison with other treatments or placebo. Therefore, it is possible that some of the patients would have achieved PCR even without dostarlimab, or that some of them might relapse later. Another challenge is the cost and availability of dostarlimab. The drug is very expensive and not widely accessible, especially in low- and middle-income countries where most of the rectal cancer cases occur. Furthermore, dostarlimab requires regular injections and monitoring by specialized doctors and nurses, which may not be feasible in resource-limited settings. Therefore, more research is needed to find out if dostarlimab can be combined with other cheaper and simpler treatments, such as radiation therapy or oral drugs. In conclusion, dostarlimab is a new formula that has shown amazing potential to cure rectal cancer without surgery in a small group of patients. However, it is still far from being a miracle cure for all cancers or all patients. More studies are needed to confirm its safety and efficacy, to identify who can benefit from it, and to make it more affordable and accessible for everyone who needs it. What are the benefits and challenges of using the new formula? The new formula we are talking about is not a single drug or treatment, but a revolutionary approach to curing diseases by modifying the genes that cause them. This approach is called gene therapy, and it has the potential to save millions of lives by correcting or replacing faulty genes with healthy ones. Gene therapy can be used to treat a variety of diseases, such as sickle cell disease, cystic fibrosis, muscular dystrophy, and rare cancers. These diseases are caused by inherited or acquired mutations in specific genes that affect the function of cells, tissues, and organs. By using a vehicle called a vector, which can be a modified virus or another molecule, gene therapy can deliver healthy genes into the cells of patients and restore their normal function. The benefits of gene therapy are obvious. It can offer a cure or a long-term improvement for diseases that have no other effective treatments. It can also reduce the need for lifelong medication or surgery, and improve the quality of life of patients and their families. Gene therapy can also prevent the transmission of genetic diseases to future generations by correcting the mutations in reproductive cells. However, gene therapy also faces many challenges and risks. One of the main challenges is to ensure the safety and efficacy of gene therapy products. Gene therapy involves manipulating the genetic material of living cells, which can have unpredictable and potentially harmful consequences. For example, gene therapy can cause immune reactions, inflammation, infection, or cancer if the vector or the inserted gene is not well controlled or regulated. Another challenge is to deliver the gene therapy product to the right cells and tissues in the body, without affecting other healthy cells or organs. This requires precise targeting and dosing of the vector and the gene. Another challenge is to overcome the ethical and social issues that surround gene therapy. Gene therapy raises questions about who should have access to this technology, how much it should cost, and how it should be regulated. Gene therapy also poses ethical dilemmas about altering human nature, identity, and diversity. Some people may have religious or moral objections to modifying their genes or those of their children. Others may worry about the potential misuse or abuse of gene therapy for non-medical purposes, such as enhancing physical or mental traits. Gene therapy is still a relatively new and experimental field of medicine that needs more research and development before it can be widely used in clinical practice. However, it offers a promising and exciting avenue for curing diseases that were once considered incurable. By using the new formula of gene therapy, we may be able to save millions of lives and improve human health in unprecedented ways. The new formula that could save millions of lives is based on gene therapy, a revolutionary technique that can modify or replace faulty genes with healthy ones to treat, cure, or prevent diseases. Gene therapy can be used for a variety of conditions, such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. One of the most promising applications of gene therapy is for sickle cell disease, a genetic blood disorder that affects the structure and function of hemoglobin, reduces the ability of red blood cells to transport oxygen efficiently, and progresses to a chronic vascular disease that affects approximately 100,000 Americans and millions worldwide. Sickle cell disease has a disproportionate effect on the Black community. Scientists have developed a way to use CRISPR-Cas9 gene editing technology on patients' own blood stem cells to correct the mutated gene that causes the disorder. This therapy has the potential to transform sickle cell disease care by producing an accessible, curative treatment that is safer than the current therapy of stem cell transplant from a healthy bone marrow donor. However, gene therapy is not without challenges and risks. Some of the issues that need to be addressed are: how to deliver the gene therapy safely and efficiently to the target cells without causing unwanted side effects or immune reactions. How to ensure the gene therapy is stable and durable in the body and does not cause unwanted changes in other genes or cells. How to regulate the gene therapy products and ensure their quality, safety, and efficacy. How to make gene therapy affordable and accessible to all patients who need it, especially in low-resource settings. How to address the ethical, social, and legal implications of gene therapy, such as informed consent, privacy, ownership, patenting, and equity. These are some of the questions that researchers, clinicians, regulators, policymakers, patients, and society need to answer before gene therapy can become a widely available and accepted treatment option for various diseases. Gene therapy has enormous potential to save millions of lives, but it also poses significant challenges and risks that need to be carefully weighed and managed.